Not only talk about the modeling, but also sort of correlations with other avenues of research, how one can tackle this whole aspect of biosignatures. So the question, of course, becomes whether on Mars or somewhere else, what should I look for in order to find former traces of life? And we heard already this morning very nicely the discussion how difficult it is to recognize microfossils in the rock record, as these examples by Schopf have shown. And I always like to show these images, which are abiotic mineral precipitates of barium carbonate, which truly look to me at first glance at least like a cyanobacteria, like oscillatoria. So one needs to be really careful. Obviously, the same thing is the case with biomolecules, which can also form, of course, they're not biomolecules, but organic molecules under abiotic conditions, like we've heard from Biebering on the comet Chury. Now, and I'm glad that this was mentioned today as well by Franz Julian, that this is a two-sided sword. What is one thing is for the people who study biosignatures, they would, of course, prefer if the abiotic world is totally different from those structures or those molecules that are produced by life. That would make it easy to recognize a biosignature. However, on the other side, and there are many of you in this room, they're actually really happy that the abiotic world produces structures and molecules that are very close to biomolecules and so forth, because that makes it even only possible to imagine how the origin of life occurred. And that goes back to experiments like by Miller, or of course these vesicles that form abiotically if you just mix some amphiphile in water, and they form structures that look already like cells. So what is good for one is bad for the other, but this is, I think, a great venue to discuss these topics. So the question I want to get into here is it only possible to find biosignatures on Mars or ancient Earth that are microscopic? Of course not. You know that because microorganisms are plentiful, many individuals, many species, and they're able to self-organize into larger macroscopic structures through quorum sensing or other processes. And then they form very commonly a two-dimensional layered community. And however one wants to name these structures, if they're thin, you can call them a biofilm. As you can see over here on this rock in the aquarium, where there's this green film covering the rock and the fish are grazing on it. But actually, those spots that are grazed off are, of course, not sterile at that moment. There is still some biofilm on there. It's just so thin that you cannot see it. It's similar to the biofilm on your teeth that you cannot see. At least one shouldn't be able to see that biofilm on your teeth. If the community becomes more complex, more species come together, more energy is available and nutrients, you can get a very complex, thick biomat or microbial mat that can additionally form three-dimensional structures. And now, if such two-dimensional microbial communities are preserved in the geological rock record, you can call it biolaminite or many other terms. And in order to understand better what one is seeing in these Archean rocks, it makes a lot of sense to look at modern examples. So for example, here on this beautiful field trip that I took with Christoph Heubeck and Martin Hohmann back 2 years to Tunisia, we looked at these intertidal microbial mats over here. They're beautifully thick, underlain by black organic material and topped off with a cyanobacterial dominated community that is pretty much hairy on the surface due to the filamentous nature of these cyanobacteria. You see all these hair filaments reaching into the water. If you pull the sample out of the water, it's like coming out of the shower with long hair. The filaments just come down. You can look at these specimens under the SEM microscope, and you see the filamentous cyanobacteria. These very thick, rubby, carpet-like biomats you can cut out, put in the sun, let them dry. Fortunately, microbes are very hardy, so they don't die anyway. So you can bring them back to Berlin, put them back into the water, and they, like a dry sponge, suck up the water. And after a few hours, you can see how the cyanobacteria are waking up here, and the filaments are crawling out of the tuft, and they're reaching out not only to get some more light, but also to get access to nutrients. So a very mobile system. And this is, of course, good, and we'll hear more about this in the following talk by Christoph Heubeck, because you can try to correlate these structures with the geological rock record, as here in a sandstone you see these dark lamina, which are believed to be organic matter and configured in a similar shape as these tufts in the modern. So that's one avenue how to study the fossil record of life, to look at modern examples. Another possibility is, of course, to do lab experiments, because nature obviously is also very complicated. It has many species, has many changing environments. If you have a laboratory setting, you can study a single species under very confined conditions. And so we set up this circular flume, basically, in which we can grow a filamentous cyanobacteria as a single species under very controlled conditions. And what we see, basically, is what we saw in nature, that this single species cyanobacteria grows on the surfaces and forms these hairy texture, these streamers, as they're frequently called, believed to be an adaptation to gather more nutrients out of the medium. Another approach is to study these single species cyanobacterial mats under tidal conditions. So this is an aluminum box about that size with a wedge of sediment in it, and the microbes have been growing for about 2 months. And what you can see is that over time, gas domes form, gas accumulates below the biomat, lifting up the biomat, including the sediment. And this is very interesting because this shows you that the biomat has a cohesive nature and it's impermeable for gases. Otherwise, it would come out and form not these gas domes. And this leads to structures and behavior that might be indicative of life. As you can see in this time-lapse video that took a few minutes, the water is rising up, the tide is coming, and this gas dome here doesn't pop, it doesn't stay put where it is, but it is basically pushed forward by the pressure of the water, including the sediment that is bound by the biomat itself. So such properties, although you could always imagine something abiotic that could behave similar, might be useful as a biosignature. And of course, we find these gas domes also in Tunisia, much larger and much more diverse. And if the tide goes away, if the water recedes, they dry up and are nicely preserved. And that's exactly also what we see with a single species in the laboratory. After drying up the sample, these gas domes are preserved. And again, you might be able to interpret these Archean layered structures as gas domes trapped under a former biomat. So that's basically the biomat. If you have the right physicochemical conditions and the microbes help a little bit out, you can co-precipitate minerals. Commonly it's carbonate, but doesn't have to be. And then, don't take these terms too seriously, I call it here just a biofilm crust if it's very thin, like in a hot spring deposit, or you have more like a biomat crust if you have a thick photosynthetic mat, as you can see over here. Now, if these structures are preserved in the geologic record, we tend to call them a microbialite. Yeah? And the standard classical microbialites are the internally laminated ones that are called stromatolites. Now, stromatolites, already described more than 100 years ago, are very useful, but they can be misleading because many abiotic structures look like stromatolites. As you can see here, the spirite and hematite deposits over here, or the malachite, they look like stromatolites if you wouldn't know better. However, they have nothing to do with life and form abiotically and would also, of course, form on Mars, for example. However, if the microbial mat is dominated by photosynthesis and very thick and very complex, you develop more complex internal textures of these microbialites. You can then call them thrombolites or dendrolites or so forth. And one example I have been studying is in southern Chile and Patagonia, where these microbialites, these brown coral-like structures grow in the lake here and are also exposed subaerially due to a drop in lake level. And why this is interesting, that to study these thick biomats that are photosynthetic, is because due to that growth dependence on the light intensity, they develop morphologies that might be useful as a biosignature. As you see, for example, here, these multiple meter tall structures, depending on how much light they get, they grow at a different rate. So up here they grow faster, down here where it's shaded they grow less, so you end up over time with this overhanging structure, which of course a mineral shouldn't do, because at least to my knowledge I don't know a mineral that grows better in the sunshine than it does in the shade. Here you can see again this thick biomat. It's multiple centimeters thick on the top, where the light is very intense, whereas if you look down at the foot of this structure, it's very dim. The biomat is rather a thin biofilm. Growth is very reduced, and you preserve the lamination of the original biomat in the microbialite structure, and you end up with a laminated texture. So would be nice if one could grow these microbialites in the laboratory as one does with just the biomats itself. But a biomat is a one-generation thing. It still takes a few months in the laboratory. But a microbialite is a long-term accumulation of multiple generations of biomats. So that even exceeds a PhD and perhaps a lifelong career. So therefore, I decided on modeling the growth of microbialites, because in this way you can accelerate time, of course, if you have the right conditions. So what you see here is basically one result of a model run, which looks like a classical stromatolite. More or less nicely laminated, pretty massive, rounded shape, and so forth. And these structures, as I said, from the morphology perspective, are quite similar to abiotic minerals, which are generally limited by diffusion due to the ions coming to the surface. And so, this type of structure you receive if the relationship between illumination and growth, now not the growth of the biomass itself, but the growth of the minerals, is configured in such a way that, of course, if you have no light at all, you have no growth. If you have a little bit light, it increases the growth intensity. However, this is a behavior where an excess of light cannot be translated into additional growth. So it's rather either a metabolically primitive biomat, early Archean or so forth, or a very thin biofilm that doesn't have the ability to basically capture all the light and translate it into carbonates. If you, on the other hand, correlate your illumination and growth in a linear fashion, meaning that however much more light is available, you grow more and more, which might be more analogous to these thick biomats that can capture all the light and are metabolically very modern, then you end up with a more intricate, knobby, branching texture, as you can see here, whereas we've seen in Lago Sarmiento in Chile. And so in that sense, one can hypothesize that stromatolites, which already occurred in the Archean, are rather built by simple, metabolically simple, thin biofilms, whereas as evolution progressed, the biomat got thicker in the Proterozoic. We see suddenly these thrombolites and dendrolites appearing. And with that, I want to thank a bunch of people, but first and foremost Christoph Heubeck and Martin Hohmann, who let me take part in their Archean endeavors, and of course down here to the left, the co-author of this talk, Matthias Müller, who is helping me writing the code for this computer simulation. Thank you.